Hello, my name is David.
And my name is Florence.
We are TeamASAP, working to transform the world at scale. Now imagine a future where large-scale macroalgae farms grow in the ocean, producing sustainable biofuels, food, and valuable resources, all with minimal labor and maximum efficiency. Welcome to the Automated Sporophyte Algae Production System, or ASAP. Our mission is to revolutionize macroalgae cultivation through automation and genetic optimization. Macroalgae farming struggles with harsh conditions, inconsistent yields, and high costs. Unlike terrestrial crops with optimized GMO seeds, it lacks standardized, scalable cultivars.
To solve these challenges, ASAP aims to automate and optimize the entire macroalgae production process. Our system has 5 key components: cultivar enhancement, which uses CRISPR-based gene editing for higher yields; micropropagation, allowing us to rapidly produce healthy gametes in a lab setting; cryopreservation, giving long-term genetic storage for consistency and scalability; group insertion, where we use dye electrophoresis for uniform attachment; and SCADA-based cultivation, automating growth conditions with sensors and AI. Each component works together to make macroalgae farming more efficient, scalable, and sustainable. In this project, we focus on the cultivar enhancement and micropropagation components.
To make macroalgae farming more productive, we need to improve growth rates, lipid content, and environmental resilience at the genetic level. That's where the cultivar enhancement process with CRISPR comes in. We are targeting gametophytes, which are easier to edit than sporophytes with tough cell walls. For our model species, we selected Nereocystis luteina and obtained source tissues from Dr. Charles O'Kelly. To induce gametophyte production, we use low light intensity, low temperatures, and F2 growth media to create optimal growth conditions.
We targeted the APT gene in N. leukemia as a counter-selectable marker. APT converts 2-fluoroadenine into a toxin, killing cells. Knocking it out allows us to screen CRISPR-edited cells that survive 2FA exposure.
Although the N. leukemia genome is sequenced, which allowed us to skip the sequencing step, the genome is not annotated. We had to conduct a bioinformatics analysis with tBLAST-N and Clusto-Omega to identify conserved regions in the APT gene across different related species. The alignment highlights conserved regions, which helped us map the APT gene structure in N. leukemia. Through this analysis, we identified 5 high-scoring coding regions corresponding to the 5 expected exon boundaries. This was crucial in selecting a precise target for CRISPR modification. Using the tool CRISPR, we designed a high specificity guide RNA targeting exon 1 of the APT gene. With this foundation of bioinformatics, the next step will be testing transformation methods to introduce CRISPR constructs into gametophytes and also validating edits with the 2FA selection. We were not able to test the experimental verification due to time constraints.
Following the cultivar optimization, the next step is mass-producing sporophytes. Our micropropagation process uses tissue culture techniques to grow new algae from small samples, increasing scalability. We tested micropropagation using dulcet algae in Mishra-Scoop medium with kinetin, sterilized through autoclaving. Algal pieces were cut, cleaned with 10% alcohol and bleach, then placed in nutrient media. Finally, cultures were incubated under a 12-hour light cycle to assess growth.
After 2 months, most samples lost color and failed to grow, but 2 out of 10 managed to retain color with slight callus growth. Failures may have been due to stress from media transplantation or nutrient and sterilization conditions, showing that further optimization is needed.
Through our work, we've taken the first steps towards scalable macroalgae farming. We successfully designed CRISPR gene edits, identified key micropropagation challenges, and laid the groundwork for automating the cultivation process.
CRISPR-enhanced algae grow faster, stronger, and richer in biofuels, making them ideal for energy, food, and bioplastics. With micropropagation and automation, we can also cut labor costs and improve efficiency. For industry adoption, long-term plans include industry partnerships, regulatory approvals, and future pilot farms for large-scale adoption. Overall, ASAP is set to transform macroalgae farming into a scalable, cost-effective industry, just like GMO crops, where farmers can order optimized sporophytes for consistent yields and lower costs. Thank you.